Hi class, we are going to go over the electron transport chain. You should have gotten the kind of legal page diagram of the electron transport chains from the supply, and then we're going to work through the video today. So if we remember from before March break, we did glycolysis, Krebs cycle, pyruvate oxidation. At the end of the Krebs cycle, most of the energy was stored in NADH, as well as there was a little bit stored in FADH2 because we had taken all the energy out of the glucose and then recycle those carbons back into that oxaloacetate to keep the Krebs cycle going. So now we are able to use those NADH and FADH2 molecules in what's called the electron transport chain to finally make those ATP molecules that the other steps have been kind of leading up to. So where does the electron transport chain happen? It happens in the inner membrane folds of the mitochondria. Kind of if you think of like in the intestines when you learned about in grade 11, it kind of folds in and out like your villi and your microvilli. Kind of, it just allows for a greater surface area. Okay, the NADH and the FADH2, as I said, are going to be used here in order to give up their electrons. And so if you think of the redox reactions, they are going to lose the electrons. And if we think of the LEO, it means that they are being oxidized. The one thing to kind of we haven't really talked about is when you think of glycolysis, there was the NADH that was produced in that process, but it's happening in the cytoplasm. The NADH molecule can't move across the mitochondrial membrane, and so it has to undergo what's called a shuttle. So what happens is the electrons from NADH gives the electrons to an FAD molecule to produce FADH2. So kind of how this looks is that you have the NADH up here from glycolysis. It's going to transfer its electrons through this glycerol phosphate molecule shuttle and become NAD plus that can be used in glycolysis. At the same time, it produces that FADH2 and the electrons are just going to move through. This FADH2 molecule can then be part of the electron transport chain. This matters in our energy count later on when we get there, just so you're kind of aware. So realize that that NADH from glycolysis isn't used directly, rather it shuttles its electrons through the glycerol phosphate shuttle to create FADH2. The ATP that we've seen produced in the Krebs cycle as well as glycolysis was through that substrate level phosphorylation. We're now going to bring in that oxidative phosphorylation because we really haven't seen it yet. So the idea is that it undergoes oxidation reactions of NADH2, NADH and FADH2 to pump electrons, hydrogen ions through and electrons through the series of proteins that create your electrochemical gradient. That is what oxidative phosphorylation is. It's the oxidation of NADH and FADH2 to create a concentration gradient through moving hydrogen ions from one side of the inner mitochondrial membrane to the other side. It also, because you get the movement of the electrons and the hydrogen ion, it's called your chemiosmosis. So you can see here, you're pumping those hydrogen ions across. That's what's creating your concentration gradient, leading to your oxidative phosphorylation to create your energy. So the oxidation is of your NADH and then your FADH2, and then the phosphorylation is in the step that's not in this photo that produces your ATP. Okay, so we're going to kind of run through what happens at each of these enzymes here. Okay, and we're going to go through the names which you need to know. You should have kind of the big long page out and then pause the video as you need to fill in the big long page. Now that you have the long electron transport chain page in front of you, also sometimes colors help figure this out. Okay, so step one. What's going to happen in step one? You're going to pause the video when you need to, copy step one under the step one box, and then we'll add into the diagram. So just so we kind of know what's going on. In step one, NADH undergoes an oxidation reaction to give up its electrons to the enzyme NADH dehydrogenase. See how it's got the A's on the end, so we know it's the enzyme. when the electrons move through NADH dehydrogenase enzyme, it causes an attraction with the hydrogen ions in the mitochondrial matrix, but the protein in the enzyme doesn't allow for them to actually come in contact with each other. So it causes the 4 protons, so your hydrogen ions, to be pumped across from the matrix to the inner membrane space. So the next kind of slide will show you what that means. So right here, this is just showing you that it's the mitochondria. Nothing crazy. This is now a zoomed in portion of here. Okay, so just so you don't realize what's going on. The NADH dehydrogenase is the pink protein here. In step 1, NADH undergoes your oxidation reaction to produce NAD+. So you can see the 2 electrons that it donated. What happens is that they're able to move through this enzyme and going to be shuttled through this whole process. At the same time, these 4 hydrogen ions get pumped across. The reason they are not able to touch those electrons has to do with the pathways. They just can't come in contact with each other the way the protein is designed. That's your step 1. Oxidation of NADH, and then the 4 hydrogen ions are pumped through. That's your step 1. Step 2 is kind of moving through electron carrier as well as another reaction happening. So those electrons move from NADH dehydrogenase through what's called coenzyme Q or ubiquitin. Okay, that coenzyme Q shuttles the electrons from NADH dehydrogenase to cytochrome BC1 complex. So it's kind of moving through so that coenzyme Q can kind of move back and forth. That coenzyme Q also has FADH2 undergo oxidation reaction to donate its electrons as well to that coenzyme Q, and then carries those other electrons. This is going to allow for us to pump more hydrogen ions at the next step. Okay, so looking at step 2, you have your coenzyme Q, cytochrome BC1 complex. We're not really going to use that much till step 3, but it's there. It's this blue, purpley colored protein enzyme. The electrons are going to move from your NADH dehydrogenase that we talked about in step 1 over through this coenzyme Q, which can move back and forth to your cytochrome BC1 complex. So you have the electrons that are moving through. At the same time, you are oxidizing FADH2 to produce FADA and giving more electron energy to the original electrons. Okay, step 3. As the electrons come into contact with the cytochrome BC1 complex, it causes that attraction between those protons. So 4 more protons, and remember the protons are going to be the hydrogen ions, are going to be pumped from the mitochondrial matrix into the intermembrane space. So we'll kind of highlight your step 3. So you have your cytochrome BC1 complex, which is the purple one, as I said in the other slide. You got your electrons that are kind of moving through here, and then your hydrogen ions that are going to be pumped through. Just going to add in. So you have your 4 hydrogen ions that are pumped through. The next step, step 4, okay, they get moved, the electrons, those original electrons that are moving through, goes through cytochrome C. That's your shuttle, so it's just moving it between the 2 proteins and ends up at your cytochrome oxidase complex. So when you're looking, you have your cytochrome oxidase complex, which is the blue one here. You have your cytochrome C, which is our shuttle. So it has the ability to move the electrons back and forth. So you can see here, the electrons are moving from the original location, secondary location, and the cytochrome BC1 to the cytochrome oxidase complex. That's your step 4. Step 5, we're going to add a little bit more to it. Okay, Now that the electrons are in that cytochrome oxidase complex, 2 more protons, and again, remember protons are hydrogen ions, are going to be pumped across from the inner into the inner membrane space from the mitochondrial matrix. So now if we think about it, I'm going to turn to the next slide and come back to these numbers. We had 4 here, 4 here, 2 here. So now I have a total of 10 protons that have been pumped across. 6 from NADH, which we need these numbers for later, and 4 due to FADH2. So these numbers here are going to matter in our final energy count, which is the note we'll do next. Okay? So while you are labeling step 5, you should have, okay, all you should show is that the hydrogen ions are getting pumped in. We still have these electrons that are here that we're going to deal with shortly in step 6. So step 6 is now still in the cytochrome oxidase complex. The electrons are going to attach to what's called their final acceptor. It's an oxygen molecule. The oxygen molecule is going to bind with the electrons, but also bind with hydrogen ions that are found in the mitochondrial matrix. And that oxygen and hydrogen creates our water molecule. So now we have those final electrons moving through, reacting to create our water. The reason is because oxygen is relatively highly electronegativity and it pulls the electrons through. So as the electrons are moving through this entire electron transport chain, they do lose some energy. So they need something that's able to pull it strong enough. And that's where the oxygen comes to play. And this is why when you take a breath in, you're bringing in this oxygen molecule because this is where it's needed for cellular respiration. So what do we have so far? Okay, we have a buildup of the hydrogen ions in the inner membrane space. I've now created that electrochemical gradient. So I have this chemiosmosis happening because now I have that, as I said, electrochemical gradient, which is a difference of concentration and charge. Because you have a buildup of a lot of hydrogen ions. So it's not just a high concentration of hydrogen, but it's a high positive charge as well. Okay. The hydrogen ions don't have the ability to go through the phospholipid membrane because it just doesn't allow it because it's on that hydrophobic, hydrophilic head and tail that we learned about in the first unit. So what it has to do is it passes through this proton channel, which is called ATP synthase. How this ATP synthase works is it turns. It has the ability to take in 2 hydrogen ion, and then when it does that, it turns, and while it's turning, it pumps out an ATP molecule. Okay, so as the hydrogen ion moves through it, it allows for this ATP synthase to move, generating ATP from ADP, if you remember, adenosine triphosphate, and you have adenosine diphosphate, and then you add another phosphate. So what you end up creating is you'll have your ADP, so your diphosphate, plus that phosphate, which would give you your ATP. So that's what's created through here. This is the process of oxidative phosphorylation, using a series of oxidative reactions to create your phosphate. Okay, so those redox reactions that we talked about in the first unit come into play here. So you can see that you have your electrochemical gradient created from those series of reactions we talked about and moving those hydrogen ions through with the help of the electron in the first part of the electron transfer chain. This is your ATP synthase enzyme. 2 hydrogen ions come through to create 1 molecule of ATP. So as this moves through, it turns and it twists. So on your kind of long page, you can add your ATP synthase to 2 hydrogen ions that are going to move through, creating ATP from your adenosine diphosphate and your lone phosphate. So now we had 2 electrons from both NADH and FADH donated to the electron transport chain to create that electrochemical gradients. Okay, so we have your oxidation reduction reactions. realize as we're doing it, some of the energy is lost at heat. So it's never going to be a perfect system with producing as much ATP as we want because there's always going to be a little bit of waste. There's also going to have kind of a leaky membrane, so some of it's going to move through as well. Okay. When we look through the reaction, you want to remember that the last electron acceptor is oxygen, where the initial electron donor is going to be NADH and then FADH2. So with that final electron acceptor being oxygen, this is what creates water where it gets released as waste product through sweat or urine or recycled. So depending what we want. Okay. So we were able to create ATP through your chemiosmosis and your oxidative phosphorylation.